The same factors that influence all living things influence the growth and survival of microorganisms. Some of these factors are also used in classification of microorganisms. Factors that are inherent characteristics of the food are called intrinsic factors, whereas factors that are external to the foods are extrinsic factors. For example, the pH of a food would be an intrinsic factor, while the storage temperature would be extrinsic. FATTOM is an acronym used to describe the factors that affect microbial growth. These factors are food, acidity, temperature, time, oxygen, and moisture. We will look at these in more detail. The first factor we will focus on is food or nutrients. An appropriate or ample food source is a critical factor affecting microbial growth. These include sources of protein, sugars or carbohydrates, and other small amounts of vitamins and minerals. Some bacteria, such as lactic acid bacteria, require sugar to grow. Many yeasts require sugar in the absence of oxygen, but will use acids such as acetic or lactic acid when oxygen is present. Practically all molds require oxygen and will grow on sugars and a variety of acids, alcohols, and other organic compounds. Control of nutrients is very rarely used as a preservation technique. The second factor we'll focus on is acidity and pH. The pH of a bacteria's environment affects both its rate of growth and its viability. Externally, a change in pH can compromise structural proteins in the bacterial cell wall and membranes, as well as critical membrane-bound nutrients transport systems. Within the bacterial cell, exposure to an adverse pH will disrupt the cell's enzyme-regulated metabolic processes, decreasing the cell's growth rate. Acidification of the cell's interior disrupts the cell's ability to generate energy, resulting in death. In acidified foods, the addition of organic acids is frequently used to reduce the pH and control bacterial growth. Organic acids commonly used as preservatives, including acetic acid, propionic acid, lactic acid, citric acid, sorbic acid, and benzoic acid. The third factor we'll focus on is temperature. Temperature may be the most important extrinsic or environmental factor affecting microbial growth. Because environmental temperatures can control the types of organisms able to grow and survive on food product, we classify organisms by their temperature requirements. In general, microbes grow slower at lower temperatures. Organisms are classified based on the ranges of temperatures that promote optimum growth. Temperatures outside this range, both above and below, will have a negative effect on growth, but will not cause their growth to completely cease. Most bacteria prefer intermediate temperatures of 85 to 115 degrees Fahrenheit. Yeasts and molds prefer lower temperatures of growth, with the optimum temperature near 70 to 85 degrees Fahrenheit. However, a number of molds grow well at refrigeration temperatures. Most vegetated forms of mold and yeast are very sensitive to temperatures of 160 Fahrenheit and greater. Most vegetative bacterial cells are also sensitive to this pasteurization temperature, but some of them are quite resistant to high temperatures. Storing foods at refrigeration temperatures is critical to minimizing food spoilage and increasing safety from pathogens. A great deal of economic loss due to spoilage results from temperature abuse, and many foodborne disease outbreaks are subsequent to a period of temperature abuse. Alternatively, heat treatments such as cooking and pasteurization can significantly reduce bacterial numbers that cause both illness and spoilage. The fourth factor we'll focus on is time. Discussing microbial growth or death requires time to be considered. All living things require time for growth. So with food processing, the time and temperature food is held before processing can affect microbial growth. With thermal processing of acidified foods, a specific time is a critical factor in processing recommendations for the destruction of microbes at a specific temperature. The fifth factor we'll focus on is oxygen requirements. Microorganisms are often categorized by their oxygen requirements and tolerance, as the presence of oxygen can be a powerful toxin to some organisms. One reason that oxygen requirements are important is that we can limit the growth of certain bacteria by limiting the amount of oxygen in the environment. This is the idea behind modified atmosphere and vacuum packaging. Aerobic organisms cannot grow in an environment without oxygen, and so the shelf life of the reduced oxygen product is increased. However, this technique would not control the growth of anaerobic or facultative anaerobic organisms, and may even increase the growth of anaerobic pathogens like Clostridium botulinum, because there would be no competition from the normal flora of aerobic organisms. The sixth factor we'll focus on is the moisture, or amount and availability of water in a food, referred to as water activity. Microorganisms require water for their most basic biochemical and metabolic processes. Water is important for transport of nutrients, removal of waste materials, enzymatic reactions, and the synthesis of cellular materials such as proteins. In order to maintain these normal cell processes, water must be available to the cell. Nutrients must be in a soluble form to enter the microbial cell through the cell wall. Sufficient moisture is also required for this transfer. Microbial growth is not only influenced by the total water content, but also by the types of substances dissolved in the water and their concentration. Most common bacteria, including all of those of public health significance, will not grow when water activity is 0.85 or less. However, many yeasts and molds have been found to grow below this level, and some will grow down to a water activity level of 0.60. Therefore, yeasts and molds are often found to be responsible for spoilage of low water activity foods, preserved by high sugar concentrations.